Hello, my name is Ayorin Afolayan from the Global Health Research Unit for the Genomic Surveillance of Antimicrobial Resistance at the Department of Pharmaceutical Microbiology, University of Ibadan, Oyo State. I'm going to be giving a presentation on data sharing, NCBI, and strain-specific databases. I'll be guided by this outline: overview of GenBank in NCBI, submission of sequence data to GenBank, overview of strain-specific databases, and submission of data to Klebsiella species complex database. GenBank is a sequence database that houses publicly available DNA sequences, including its annotation that's been submitted by the scientific community around the world. The GenBank is part of the consortium of sequence databases that form the International Nucleotide Sequence Database Collaboration, which includes the DNA Data Bank of Japan and the European Nucleotide Archive. So it means that all submissions that have been made in GenBank has its own record at the European Nucleotide Archive and the DNA Data Bank of Japan, because the three organizations exchange data on a daily basis. So how do we search and retrieve data from the GenBank? We simply visit the Entrez Nucleotide website with this link here, and then you can, in this search bar here, you can just add the accession numbers or the particular organism that you are interested in identifying or in studying. The accession numbers simply means unique identifiers that are associated with a particular sequence data. And so, for example, here, I included the accession number on this search bar, and after clicking enter, it gave me information about this particular organism, which is a Salmonella strain from Ibadan. You can, as I said earlier on, you can include, if you're interested in, for example, Pseudomonas aeruginosa, you can just type Pseudomonas aeruginosa here at the search bar, and then you get a lot of information on the different Pseudomonas aeruginosa sequence data that has been submitted previously. You can also search and retrieve data from BLAST, which is the basic local alignment search tool, many of which could be either BLAST-N, BLAST-P, BLAST-X, T-BLAST-N, or T-BLAST-X, depending on whether you are interested in searching for similarities between your data, your sequence data, which is DNA sequence or protein sequence. That's your query data compared with the sequence data that are available already on the database to find areas of similarity. Now to the submission to GenBank. One has to register first, because after submission, you get a message from the NCBI curator saying that they've received your submission. And so they need to be able to send that message to a valid email address. So you have to register first before submission. There are two basic submission types and submission has been made easy by the NCBI. In the NCBI platform, we normally use the bank it, and it shows, this is the web platform that is shown when you click this link, this bank it link. It gives you the opportunity to select the type of sequence data you are willing to submit. For example, if you're interested in selecting, in submitting SARS-CoV-2 sequence data, then you can click this, and then and then after that you can click start, and it will guide you step by step on the procedure for submission and on which database, the right database to submit your sequences to. They also have the submission portal. When you click this link, it will bring you to this particular web page, and then you can easily search for the sequence type, sequence data type that you are interested in. So for example, if you're interested in submitting 16S ribosomal RNA, you can just click 16S ribosomal RNA, and then it will guide you step by step and lead you to the database where you are to make the right submission. There are some submission requirements. You need to have detailed metadata. For example, if you are collecting Pseudomonas aeruginosa, for example, you need to mention that you need to give information on the source. Where was it isolated from? Was it from humans or from animals? If it's from humans, which part of the human body was it isolated from? Was it within an hospital setting? Where's the geographical location? Where in Africa, or is it in Europe? Even in Africa, where exactly? So you have to give a lot of details on the geographical location, on the date of collection too. What kind of sequencer was used? And I think those are the basic information you need to give. There are some submissions that won't be accepted. For example, if the sequence length is less than 200 nucleotide, by the time you submit it, you'll get a message in a few days saying this submission cannot be accepted. Also, if you submit sequences that have contaminants, I did that one time, and then I got a message afterwards telling me that this submission cannot be accepted because this submission is a mix of genomic DNA sequences from either the same organism or two different organisms. So it has to be a submission coming out from sequence data derived from a pure culture. And then they don't also accept primer sequences or protein sequences with no underlying nucleotide submission or even non-contiguous sequences. I've submitted a couple of sequence data before, and I'm used to the sequence read archive, which is one of the databases used, and it supports the submission of unassembled data. That's raw sequence data. This is an example of the steps that you are going to take while submission. By the time you get to the submission link, you see the new submission icon. After clicking it, you are going to provide the title of your project. In my own case, it was metagenomic analysis of bacterial communities in water and soil of the Fulani and Noel Fulani in Nigeria. You are going to give a brief description. These are mandatory parameters. After giving a brief description, what's the relevance is for the medical community, or does it have environmental relevance? Then if the project was supported by a grant, you're going to give details on the grant or the funding body. You may not have details on publications yet because it's expected that you would have submitted your sequence data and have received accessions for each of your unique sequence data that has been submitted before submission to before submitting your paper before submitting your article to a particular journal, because journals always request that any study that is reporting on findings from sequence data, that's if you've derived your findings from analyzing sequence data, then you have to give details on where you've submitted the sequence data to before your paper will be accepted. And so you have the sequence accession, which I already explained earlier, that they are unique sequence of letters and numbers that are associated with a particular submission, with a particular sequence data. And you can see for me, I have up to, I think, six, yeah, 7 submissions. And you see that they are all raw sequence data, unassembled sequence data. So after approval, what next? Just to recap, you are going to get a submissions ID that you'll be sent this all of this information by the NCBI curators. You'll get about project ID, which is unique to your own project. And then you are going to get accession numbers. You can easily, it will be contained within a file and you can download it. Then you can easily manage data if you need to edit some aspects or add, just as I did previously, I added my publication to this record. So there are also strain-specific databases where one can access genotypic and genomic information for different microbial species. One of the specific databases here that I'll mention is PubMLST. I'm not going to do so much on that, but you'll find a lot of information for over 100 microbes. We also have the enterobase, which has a lot of information on about 8 microbial species, including the sequence type, CGMLSC, and a lot of information on the antimicrobial resistance and virulence information. Then the one I'll be dwelling, this database I'll be dwelling much more on the biggest DB Pasteur database, which hosts a couple of databases, including Klebsiella, Listeria, monocytogenes, Escherichia coli. It contains information on multilocal sequence typing, whole genome based typing, and other supplementary schemes. Just as I mentioned, you have information on antimicrobial resistance and virulence. So I'm focusing more on the Klebsiella pneumoniae database, the Klebsiella species complex database, which is a database for Klebsiella pneumoniae, Klebsiella quasi-pneumoniae, and Klebsiella varicola. There are 2 specific databases within the Klebsiella species complex database. We have the sequence and profile database, and we have the isolates database. So the sequence and profile database contains information on the alleles and the MLST profile, while the isolates database contains information on the genome ID and the FASTA file, that's the assembled rates. So in order to submit isolates, you'd have to request for a username and password by sending an email to the curators, the Klebsiella MLST curators at the Pasteur Institute. They would ask you if you are the PI, the principal investigator, because it's necessary for each laboratory to have just one single username and password, just in case the scientist who conducted the experiment beforehand is leaving. And but the PI would always be the PI. So they would always ask you whether you belong to a laboratory and whether you can supply the email address and name of the PI. And then that's what they will use to create the temporary username and password. So as I said earlier on, we have two databases within the Klebsiella species complex database. The sequence and profile database is one of them. And after obtaining the username and password, you log in, you get to the submissions icon, you click submissions, and then you click manage submissions. And then this will take you to a page where you have the opportunity to choose whether you want to submit alleles or MLSC profiles, as seen in the next slide. So if you're interested in submitting alleles, you have the opportunity to choose which of these loci you're interested in. We have the opportunity to choose from the loci scheme, and then from here you can choose the specific locus that you're interested in submitting. Then you choose the sequencing technology, whether it's Illumina or the 454 or whatever it is, whatever sequencer you used. You choose the read length, choose the coverage, then choose the assembly, whether it's de novo or whether you used a reference-based assembly. You choose the assembly software, and then if you have, it's advisable to include the FASTA sequence corresponding to the locus you want to submit, and then you can click submit. But if you want to submit new MLST profiles, you can just paste the profiles here. In my own case, because I did a recent submission, I was interested in submitting some MLST allele. And I selected one of them here. I had the sequence corresponding to the allele, and then I just clicked submit. And that was it. I didn't submit the profiles. If you are interested in submitting the isolates, which is actually advisable, it's not only advisable to submit the sequences corresponding to the alleles. It's also important to submit the genomes, the complete genomes, the assembled genomes rather. So when you log in, you click on the submissions icon, you click on manage submissions, and then you choose the submission type, either the isolates alone without the assembly files, or the genomes, that's the isolate records with the assembly files. It's advisable to submit the isolate records with the assembly files, at least for records. So in my own case, I submitted the isolate records with the assembly files. And so if you're only interested in submitting new isolates alone, you can either just paste in a tab delimited text or download this plain text or the Excel spreadsheet, fill it, and then upload and just click submit. If you're interested in submitting new isolates with the genome assemblies, you can also follow the aforementioned step, but you'll be given the opportunity to submit the assembled reads themselves, the assembled data themselves. And then you get information from the curators, depending on how busy they are, in a few days or a couple of weeks. And if they have any issues with your submission, they'll get in touch with you and ask you to supply further information. And if there's an error in your submission, you'd get a link saying there's an error in your submission and you need to resubmit or do some other works that would allow your submission to be accepted. And that's all. Thank you very much.